Morning and welcome everyone. I want to thank ARM for the opportunity to talk to you today about how Kytopen we are accelerating the future of medicine. Kytopen has developed a solution to the costly and lengthy cell therapy development timelines by uniquely scaling from automated high throughput discovery and optimization to large volume clinical manufacturing. This is not possible with current batch-limited viral and non-viral engineering processes today. As the industry has advanced, there are over 100 gene-edited trials going on today. Last week, we had the very exciting news from Vertex and CRISPR related to the completion of their global regulatory filing of their ex vivo cell therapy with the FDA, a first using CRISPR. But delivery remains a major bottleneck at every phase of development. Payload sizes, multi-edits, scaling, large volume production limitations exist. Not to mention the cell's innate ability to desire to defend itself against editing. All along the development process, product yields are negatively impacted due to current delivery techniques. Of those techniques, viral remains the majority, the standard in the industry. But even with all the investment in capacity over the last few years and the subtle improvements in the technology, challenges remain. The good news is that in the past few years, non-viral methods have become more and more of an option, both in terms of safety and effectiveness, and in particular in first-generation gene therapies. Most non-viral transfection solutions are based on electroporation. Static electroporation utilizes only 2 of the 3 potential energies in the process, chemical and electrical. It has limitations, though, in terms of scale, volume, and batch size. And as you increase the amount of electrical energy, you increase the risk of cell death and potential genetic dysfunction. So even with all our advances in recent years, gene delivery remains a major bottleneck throughout the therapeutic development life cycle. The serial iterative process offers limited control and continual need to reoptimize and confirm at all steps along the development process. This creates waste, expense, and takes too much time. All that adds up to a development cycle that is not sustainable, with its current cost exceeding over billions of dollars and development times well over 10 years as patients wait for therapies. Our belief is that if you start with our flow-effect array in discovery and use flow-effect along the entire therapeutic development journey, you will save significant time and money and eventually save lives. We are transforming this process by providing you more powerful tools that enable you to get answers in days and weeks instead of months or years, and allow you to optimize once with more flexibility and control, and enable you to manufacture at whatever volume you need. We accomplish this through 2 embodiments of our technology. The flow-effect array you see on the right, with now our first-generation device available through a pilot program and with access through a few CDMOs and other partners, and our flow-effect TX, our manufacturing device on the right. They both utilize our patented and unique flow cell at the heart of the transfection process. This flow cell enables continuous transfection that is gentle, flexible, and scales with time. This single flow cell is used on pipette tips for low volume, high throughput automated work on our array and in our automated enclosed manufacturing system, the TX. The TX has a dynamic volume range that scales with time. So the more cells you need, you simply increase this transfection time. So what I like to say is it's a picture of a faucet. If you're filling a cup versus a pitcher versus a bucket or a pool, you just use time. This continuous flow opens up a mechanical subsystem for transfection that enhances payload delivery kinetics and enables dozens of parameters to discover and optimize your process. The mechanical energy allows for the reduction of electrical energy required for efficient transfection while improving cell viability, in some cases by as much as 1/5 of the electrical energy required. In a closer look at our array, you can run a full plate of 96 unique conditions in less than 10 minutes. We have designed a unique DOE software that allows you to manipulate the transfection parameters and investigate CRISPR screens, cell prep variation, payload concentration, buffer optimization, donor variability, and more. You can see here, no pointer. On the y-axis, we have plotted efficiency, and on the x-axis, viability. This is 324 reactions done in 1 day's time with T cells and RNP. You can see highlighted in the upper right is a reference or a set of conditions in the region which is high efficiency and viability off of the reference standard, which is normalized across that axis. It's somewhat hard to see, but there are some X data points as well as some triangles which represent static electroporation. And you can see that many of the conditions on the upper right are with reduced electrical energy, and this can lead to further investigation if desired. Another example of the power of the discovery array is when you use multi-objective optimization. This case has for NK cells and TIGIT. You can see multiple hot zones too here that enable you to make better decisions and trade-offs of your desired process outcomes. The data is then scaled, and you can see with the graph to the right of that, confirming your process conditions done at small scale. Both of these devices can be utilized throughout the development process for engineering confirmation runs, optimizing both up and downstream parameters, and for scale-down modeling. At manufacturing, your development transfection profile is simply programmed on the FlowFect TX. A simple and easy-to-use interface walks you through a few-step process for setup, leaving you ready to transfect billions of cells in minutes. We have the flexibility in consumables with weldable tubing and lure lock connections that enable vessel-to-vessel input-to-output configurations. The device is also compatible with automation control systems such as Delta V, and you can connect directly to up and downstream cell selections and bioreactors through the weldable tubing. Here you see some data from some collaborators. This is T cells and mRNA. You can see we worked together collaboratively at a small scale, and then they took the device and confirmed scale up in their hands at process volumes of 20 mLs and 200 to 500 million cells processed. And you can see all this was done in 1 week's time. In summary, the next generation of living medicines holds great promise, but faces issues with scaling from manufacturing to discovery. From manufacturing from discovery. The gene delivery bottleneck is costly, time-consuming, and negatively impacts product and process yield at every phase of development. And the FlowFect platform dissolves these early barriers to gene delivery to reduce time and costs from discovery through manufacturing. Just some highlights over the last 6 years. Since our Series A in September of 2021, we have moved into dedicated office and lab space from our original incubator, and we now have the 2 devices the Array and the TX, to cover the full spectrum of your discovery and manufacturing needs. And thank